Hi everyone, welcome to the second part of Microbial Genetics chapter. This will cover gene regulation, mutations, gene transfer, and plasmids and transposons. Regulation of gene expression will be discussed by first defining two major sets of genes, constitutive genes and adaptive genes. Constitutive genes are genes that can be described as always expressed. Most of the genes in a cell would be classified in this category of constitutive. Mainly, these genes are involved in metabolism-based needs of the cell, which exist as something that then are ongoing events throughout the cell's life. For events that don't happen as often or are not occurring on an ongoing basis, we have the adaptive genes, which exist in cells that then are genes that are expressed only on a needed basis. So the idea is to turn off or on certain gene activity when appropriate. And this helps to save energy. The goal of adaptive genes is to help the cell so that it doesn't waste energy during times when it doesn't need those genes. A good example of that would be the lactose operon, which will be discussed in a couple of slides. The lactose operon and other such operons contain genes that are adaptive so that they're only expressed, which we can also refer to as turned on, during some sort of stimulatory event. So something changes in the environment and it stimulates a cell to turn on these genes and then take advantage of a new situation. So we'll write down here the example, lactose operon. And the way that the lactose operon works is that it only turns on when lactose is present. And before we get to the lactose operon, let's take a look at something called phase variation. Phase variation is a term used to describe adaptive genes being turned on or off, and those adaptive genes are often contained in a complement of genes that we refer to as an operon, as we'll see in the next slide. And when an operon is turned on, it will lead to an observable or phenotypic event that we can see. This phenomenon of phase variation is something that occurs from parent cell passed to offspring because it's genetically based, and certain stimuli in the environment will then cause regulatory proteins to turn these genes on or off. A couple of popular examples listed here include very obvious changes that we can observe. So Neisseria gonorrhoeae can produce Fimbriae while they attach to the urinary tract or to genital mucous membranes when they sense that they're in that environment. Streptococcus pneumoniae similarly can produce a capsule in order to attach, say, to lung tissue, and these events can be turned on at the right time when the bacteria sense that there's a reason for them to produce these attachment mechanisms. So let's look at an operon. To begin understanding the operon, let's start with the technical definition. An operon is a regulatory unit of DNA that includes a promoter, and structural genes. The three elements that are listed as promoter, operator, and structural genes are located in the control region here for the promoter and operator. The promoter is first in the operon as it's ordered. The operator is downstream of the promoter. And then you have the structural genes, Z, Y, and A. Structural genes can be labeled by different letters of the alphabet. In this particular diagram, the Z, Y, and the A structural genes represent functional protein products that can be produced when the operon is turned on. The control region acts like an on-off switch to control whether the structural genes have the opportunity to produce their protein products. Outside of the operon, we have something called the regulatory gene. And strangely, the regulatory gene here to the left of the control region has the letter I. And that I is in reference to what we call the inhibitor. So I is in reference to inhibitor, and what the inhibitor is a protein product that comes from this I regulatory gene. So the inhibitor protein is also referred to as a repressor, as we'll see on the next slide when we start to examine the specifics of the lactose operon as a model. Here we can gain a much more specific understanding of how operons work by looking specifically at the lactose operon. Here in the lactose operon, we can see that the promoter region has something called RNA polymerase bound to it. RNA polymerase is an enzymatic complex that will transcribe the DNA, specifically the structural genes, in the lactose operon, but only if the operator region is clear. Here's where the operator region is. Let me go ahead and label that with the letter O. It was missing there. All right. We can see that there's something different about the operator region, though, in that a repressor protein is bound to it, and it's blocking RNA polymerase from proceeding through the control region, where the promoter and operator region are, and then this is now inhibiting or blocking the transcription of the structural genes. So the structural genes are blocked at the present moment from undergoing energy when the cell is at rest, when there is no lactose to be digested. Transcription. All right, now also we should take a look at the inhibitory or regulatory gene here where the letter I is. So in this case, let's keep in mind that because that's outside of the control of the lactose operon, we can label it as always on. And if you recall from a couple of slides ago, or the first slide, we defined the term constitutive as being always expressed. for having activity. And so it's important to understand that this letter I for the regulatory gene, the inhibitor, is not under the control of the operon. Not under operon control. So what happens is this letter I gets transcribed, and then what we have down here is a repressor protein that is made as a result. And that repressor protein is in an active conformation that can bind to the operator region and block transcription. Now let's look at what happens when the lactose operon turns on. This process is referred to as induction, and induction occurs when lactose is present. So let's say lactose enters into the cell because there's lactose in the environment, and a little bit of it can diffuse across the cell membrane of a bacterial cell, and then when that lactose floats into the cell, something will occur where the lactose actually will get changed into something called allolactose. Allolactose is known as the inducer, and this little molecule that results from lactose diffusing into the cell will then bind to the repressor protein. So the repressor that we saw that was being produced in the last slide, that repressor protein will change its shape so that it enters into what's called an inactive state. And this inactive state will not be able to bind any longer to the operator region. So what we see is that the operator region is now open, and the RNA polymerase that's way over here has had a chance to exit this region and successfully transcribe structural genes. When those structural genes are transcribed, eventually we'll get these functional protein products that allow for lactose digestion. One of them is beta-galactosidase, another is called permease, and yet another product is called transacetylase. Collectively, these three functional protein products will aid in lactose digestion. If you recall from laboratory, we learned that beta-galactosidase will split the lactose into two. The disaccharide will become two monosaccharides, glucose and galactose. The permease, if you guys recall from the ONPG experiment in laboratory, is a protein product that allows for lactose to be transported into the cells. And transacetylase also seems to aid somehow in lactose digestion, although the function of transacetylase has been debated quite a bit. To summarize, we'll look at the lactose operon during induction. This is when it turns on. So we can say in the sequence of events that are bulleted here, first, the regulatory gene codes for the repressor protein and is always on. Another term for that is constitutive. And the repressor protein will bind to the operator region to block transcription before lactose is present. But then during induction, which comes about when lactose is present outside the bacterial cell, some lactose will diffuse in. It turns into allolactose, and that allolactose will bind to the repressor protein and change the repressor protein so that its shape will no longer bind to the operator. So if the operator is unbound, then that means the control region will be open and it'll allow transcription to occur. I'd like to add on one other important point here, which is that glucose levels will also need to be low during the time that induction occurs. And this is because glucose is a simpler sugar, and the cell will sense glucose if it's there and preferentially break down that monosaccharide as opposed to using the lactose operon as we're studying here, because lactose is a disaccharide milk sugar. So that's an important point to keep in mind as well, is that glucose levels must also be low in order for this operon to function properly. Now let's look at a quiz question. Here's a question that could be good not only to check how can you locate the right answer, but why are the wrong answers wrong? So it reads, the lac operon is, now let's look at the first choice. Is the lac operon always on? And we would say no, because remember, the concept of an operon is that it can turn on or off so as to save energy when it doesn't need to be used. So always on doesn't make sense here. All right, let's look at the next choice. The lac operon is it on when glucose levels are high and lactose levels are low? If you recall from the previous slide, this is the opposite of when the lactose operon functions. All right, how about thirdly, the green answer. Is the lactose operon on when glucose levels are low and sucrose levels are high? This particular answer is wrong because sucrose is not involved at all. So that answer does not make sense. How about the fourth choice? The lac operon is on when glucose levels are low and lactose levels are high. And you got it if you recognized that this is how the lactose operon would function. Lastly, though, we should also understand that always off is a wrong answer, because the lactose operon fundamentally has to be able to turn on or off. So I hope you understood the correct answer here. Let's move on. Now that we've talked about how a cell can turn itself on, say in the presence of lactose, as in the lactose operon, which is a sort of a positive feedback that the cell can receive, let's look at the opposite. We're going to turn things around and look at something called feedback repression. This is a phenomenon that's outlined here in this set of bullet points, in which it states that we could have an excess end product of some sort that would build up in a cell and suppress the genes, the genetic apparatus involved in what those genes produce, which in this case is described as enzymes that are produced. And feedback repression, by comparison to, say, the lactose operon, what it will do is it'll alter the production of a cell's enzymes or protein products, but not necessarily the activity. So when we look at feedback repression as a phenomenon, we can say that it is slow and has long-term, longer-lasting effects. And we're going to look at a model for feedback repression called the tryptophan operon. And so here we're talking about tryptophan. If you recall from laboratory, that's an amino acid that some bacteria can use as a food source, as a nutrient, or if they do not have that food source, there are cells that can actually make their own tryptophan amino acid. So it would be one of the non-essential amino acids that they can make for themselves when necessary. And the genes that synthesize tryptophan have to be turned on when those needs are present. So let's look at the tryptophan operon and see how it models. It works as a model for feedback repression. Okay, so this should look familiar in the sense that we can recognize this setup as an operon. The structural genes are a little bit different though. We have E, D, C, B, A. Try not to worry too much about what the letters of the alphabet mean, and the fact that they're out of alphabetical order has mostly to do with the order of their discovery. But again, those are technical details that we won't have to worry about. What we want to understand here is that we're starting with a picture where tryptophan is in low concentrations inside of a cell to begin with. And if the cell has the ability to make this amino acid, then it will turn on. It'll start with the tryptophan Operon genes ready to transcribe and translate protein products, enzymes, etc. that will make the tryptophan amino acid. And so we look at the tryptophan Operon as a way of regulating gene expression to make as much tryptophan as necessary. And then once it has exceeded its needs for tryptophan, it will turn off the tryptophan Operon. So we're looking at it to begin with when its needs have not been met, and it'll make tryptophan until it no longer needs tryptophan. Now, this is what happens when enough tryptophan has been made. This is the sort of the flip side of the situation. Tryptophan is represented by this little molecular structure here, bicyclic ring, 6-membered ring connected to a 5-membered ring. When enough tryptophan is made, then the cell will have enough amino acid, tryptophan, that will then bind to the repressor protein, which in this case starts out in the inactive form. So I should label that. Okay, we're going to label this as an inactive repressor protein. And the inactive repressor protein is produced by the I gene that is outside of the control of the operon, right? That's a constitutive gene, just like we saw in the lactose operon, and its function is to make repressor proteins, and eventually those repressor proteins will go from an inactive state, which is opposite how the lactose operon was, into an active state when they make contact with excess tryptophan. When excess tryptophan amino acid builds up, and these repressor proteins will become active. And when they become active, they'll bind to the operator region here and block it so that the RNA polymerase does not have a way to proceed through and transcribe the structural genes E, D, C, B, and A. And that would be what we call feedback repression that's occurring in this picture. So in summary, for the tryptophan operon, we can say that the tryptophan operon produces repressor proteins that cannot bind to the operator initially. The repressor proteins are inactive and transcription will occur. But when enough tryptophan gets made by allowing the tryptophan operon to operate until more than enough tryptophan has been made, then the tryptophan levels will build up to the point where that excess unused tryptophan will bind to the repressor protein and form a complex that will bind to the operator region. And what happens is then it shuts down the tryptophan operon, and transcription will be blocked at that point. So it shuts down production of tryptophan when the cell no longer needs tryptophan. Now that we've covered feedback repression I want to compare it to an earlier concept that you learned in your metabolism chapter. This slide entitled Feedback Inhibition was copied and pasted from the metabolism chapter. And in review, feedback inhibition sounds similar in its wording to feedback repression, but inhibition and repression have two different meanings in terms of cellular function and cellular genetics. Feedback inhibition, if you recall, is a scenario where a cell will use a certain enzymatic pathway until products of that enzymatic pathway, end products, build up at the end, and as they build up, excess end product will bind to the allosteric site on the first enzyme of that pathway. All right, so we call this an end product buildup, which causes something called feedback inhibition to occur. And the way that the inhibition works is that the active site on the first enzyme of that pathway will deform. It will stop that pathway from continuing to go down in progression to continue functioning, and the pathway will shut down. And once the pathway has shut down, it will stay shut down temporarily until the end product that it was once producing, its levels may fall as the cell uses up the end product, and then eventually the active sites of the enzyme that controlled that pathway will reform and the pathway will resume. And we would call this like sort of a quick on-off switch, so to speak. It's quick, it's short-term. It's like telling a set of factory workers to simply go on lunch break and then come back when they're done. You haven't fired the workers. You're just simply telling them to stop activity for a temporary amount of time, but then they can come back. This compares very differently to feedback repression. Feedback repression would be a version of telling the workers, your job is done and we don't know when we want you back. In fact, you can just go away. So you would have to rehire workers, recreate a workforce in feedback repression. Feedback inhibition, on the other hand, is just like telling your workers to take a break. So these are two very different levels of activity in a cell in terms of how long the effects last and what it takes to resume activity again. So by comparison, feedback inhibition, as we just reviewed, affects enzymatic activity through something called allosteric inhibition. It's fast-acting and has short-lived effects. However, feedback repression does not simply affect enzymatic activity. Feedback repression actually affects enzyme production. And so that's like saying, well, do we produce these workers, do we hire these workers or not? So if you shut down enzyme production, then the enzymes essentially start to go away. And because feedback repression works by shutting down or affecting enzyme production, this is going to have a slow acting type of way in which it works. All right, slower acting. And because of that, it's going to have longer term effects as well. It is not easy to resume making enzymes once you've taken them away. It's going to take time to build those enzymes back up. And so when we compare feedback inhibition to feedback repression, we see this different dynamic here. So it's good to keep that in mind. Two very different ways of regulating a cell's activity. Now let's go on to a different subject, one about mutation. Mutation is defined most simply as a change in genetic material. All right, so the DNA has some sort of lasting change, and that DNA, when it changes, may bring about neutral, beneficial, or harmful effects. Mutations are unpredictable. There's not always a way of knowing or predicting when the DNA changes what it will do to a cell in terms of the cell's quality of life, or in fact if the mutation could even be deadly for that matter. A mutagen is a chemical or an agent of some sort that can cause changes in the DNA, bring about mutations. And so mutagens are oftentimes referred to as carcinogens in our environment when we speak about what can happen with respect to cancer in animals and in humans when certain chemicals bring about mutations. However, even when mutagens are not in the environment, it is possible for spontaneous mutation to occur just because sometimes nature makes mistakes with its DNA polymerase. So we can say that the DNA producing enzyme called DNA polymerase may randomly make mistakes. A random error may occur, and this would be what's called a spontaneous mutation. And it doesn't happen that often, but it can occur, and we call this a spontaneous mutation. Mutations can be categorized in many different ways. One such way are what are called base substitutions. This is where a mutation could result in a change of a base pair. So a single base pair illustrate, if you had a base like A for adenine, we know that A bonds with T across from it. So adenine would bond with thymine, but then let's say that a base substitution occurs. Maybe the letter A is undergoes mutation, and what if it became a letter C? So if A became C, then C would end up pairing eventually with a G rather than a T, and so maybe an AT pair becomes a CG pair, and this would be an example of a base substitution. Now, whether that base substitution causes any harm is another factor in which we can take into account. So silent mutations would be mutations where no harm was done. We would also describe it as a neutral type of mutation. And silent mutations can occur where a base changes, but the codon, the resulting codon, still codes for the same amino acid as before. Okay? And a second type of base substitution, which can result in much more serious consequences, are what are called missense mutations. So a missense mutation is where the base changes, but the codon is different. In missense mutations, you would say a base change and that change then results in a different codon. that codes for a different amino acid. An example of a missense mutation that we're well aware of in human genetics is the cause of this condition that we see here to the right. These are some red blood cells, and this particular one that has that crescent shape is a result of a missense mutation. And so one single codon changed, sorry, I put different codon, a different amino acid results, and this can cause, this is the cause then of what's called sickle cell anemia. And thirdly, we have deletion mutations, also referred to as nonsense mutations, and these are arguably the most serious, and in fact, in most cases would be lethal. A deletion mutation is where an unexpected stop codon pops up. So a base change results in an early or random stop codon. And this results in a protein fragment. A protein fragment then is truncated or not full length. One such example of a nonsense mutation that was not lethal but has been causes great handicap in its victims is cystic fibrosis. So people who suffer from cystic fibrosis have a deletion mutation in their chloride secretion channels. If you recall from an earlier lecture in micro, we talked about how their secretions are abnormal, and it's actually a result of a deletion mutation, interestingly enough. A second major type of mutation is a class of mutation called a frameshift mutation. And it's defined as an insertion or deletion of one or more base pairs in the DNA. The ultimate effect of this is that it causes a change in the reading frame. And what that means in the DNA is that all the codons that occur after the change in the reading frame are going to be garbage. We'll see that on the next slide, where I have an example of what it looks like when the reading frame changes. But ultimately, many amino acids will be coded for incorrectly. In this slide, we can look at understanding the mutations in summary by using English language in place of ordinary codons. So in genetic speak, the A's, G's, U's, and C's that are in the RNA define every 3 letters as a codon. But if we use English language, we can start with wild type, which would be an unmutated genetic original message. And it reads, "The big bad dog ate the fat red cat." Now, when we look at mutations that can occur, it should be a lot more intuitive. Substitution and deletion mutations will then result in things like, "The big bad dog ate the fit red cat." So we get a substitution, fat for fit, and it changes the meaning. So we would call that a missense, right? It's no longer the same. It is a missense. Now a deletion mutation, this on the other hand is the big bad and then no more message, because a nonsense codon is put in and it creates an unexpected stop. So we get the so-called protein fragment that was described in the previous slide, or 2 slides ago. And so we get a protein fragment, also called a truncated protein, like in the example of the cystic fibrosis patients who have compromised chloride secretion channels. And then lastly, we have frameshift mutations as an example. And in this case, if you insert a letter in the middle here, so we see after the big where it's bad, insert a B in between the A and the D, and now you have the big bad. All right, so there's your insertion. And you can see how it changes the reading frame, and everything after it is garbage. Alternatively, we have a deletion, and in this case, a letter A is missing, and it changes the reading frame, and disastrous results occur. Once again, we get something that can no longer be made into a meaningful product. Here's a figure copied and pasted from the book that helps to summarize everything that I just mentioned in a nice table for reference. You don't have to memorize this table, but it does give you a chance to see about, say, where it says normal gene. Remember, this is what can also be referred to as the wild type. And we also have then examples of missense mutations, silent mutations, as well as frameshift mutations, insertions, or deletions. The frequency of mutation can also be talked about statistically in genetics. So I had mentioned at the beginning of this section that spontaneous mutation will occur when DNA polymerase makes a random mistake once in a while. And it's estimated that this only happens 1 in 10 to the 9th replicated base pairs. So 1 every billion bases that gets made, DNA polymerase will randomly make a mistake. Randomly, spontaneously. And then we have the other possibility, which happens much more often, but it's beyond natural. This is when, in terms of exposure to certain chemicals or agents, can then influence the DNA or the genes to mutate much more often. So we can talk about how most mutagens are suspected to be carcinogens or causes of cancer because of their ability to cause damage or mutations in the DNA at a much higher frequency than would be present under normal circumstances. Not all mutagens are carcinogens, but many of them are. Keep in mind, in the human body, we have our liver and ways of detoxifying some of these mutagens. But depending on the exposure level and then the action of such mutagens, cancer can pop up. All right, let's do a concept check, and then we're going to move on to some of the examples of potential mutagens or carcinogens. So this question reads, which of the following types of mutations will have the most devastating effect on a cell? So what are our answers? We have point mutation, frameshift mutation, missense, nonsense, silent. Well, this would almost be a good essay question just to ask you to write, well, according to your understanding, which kind of mutation do you think would be most devastating? I'm going to go with the nonsense mutation, because a stop codon will actually be inserted and the cell will have the least amount of chance to produce anything meaningful because it will have been stopped short. So this is a randomly created stop codon, if you recall. It gives the cell the least amount of chance to do what it's normally supposed to do. All right, moving on. Let's look at the specific examples of mutagens. Ionizing radiation. Now, this would be an example of how a mutagen, which will then mutate the DNA, does not have to necessarily be a chemical per se. Ionizing radiation includes X-rays and gamma rays, which can pass right through the human body, but without there being an exposure directly to a chemical. So an X-ray, we're familiar with that. You go in, it can go right through, it takes a picture of your bones, and as it travels through your body, there's at least a risk that it can cause ions to randomly form within the cytoplasm of the cell, and those can react with the DNA and cause damage. So too many X-rays would be bad for you. Of course, gamma rays are even more powerful, and again, exposure to such powerful radiation will damage the DNA, maybe so greatly that it's difficult for our bodies to recover. And so that's one class of mutagen. A less powerful mutagen, but one that also is still can be incredibly dangerous, are UV light mutagens, where the form of radiation is non-ionizing. It doesn't travel far below the human skin or into an organism, but on those surface cells and the cells just below the surface, UV radiation will get in, and instead of ionizing the cytoplasmic contents, what it does is it can strike the DNA within the cell and most often cause adjacent thymines to dimerize. There are two such thymine-thymine dimers that can occur as a result of UV light exposure. We don't have to memorize them for this course, but I would like you to at least take note that UV does most often result in thymine-thymine dimers as a form of mutation. And if you get too many of these thymine dimers, it can lead to skin cancer. And so we do see the connection that too much damage to the DNA can lead to mutations that, if they're lasting and they don't get repaired, can lead to cancer. And so that is another type of mutation that we need to know about. In bacteria, bacteria cannot get cancer, but they can just simply die as a result of too many thymine dimer mutations. So UV light will kill bacteria if given enough exposure in time. There is a way to repair thymine dimers through an enzyme that humans have called photolyase, and also some bacteria. E. coli, for instance, is known to have photolyase repair enzymes, and these photolyase enzyme repair mechanisms are able to recognize enzymatically a thymine dimer, bind to it, and then undo the dimerization. In the absence of photolyase, or in addition to photolyase, there are also what are called nucleotide excision mechanisms, so-called dark repair. And this is an instance in which, beyond photolyase, which is activated by cells being exposed to sunlight, you have these backup mechanisms for repair, in which whole sections of DNA may be identified as mutated and then cut out, filled back in, and repaired through a process called nucleotide excision. The details of that were gone over in lab as well during the UV light section. So you can refer to that if you would like more information on that mechanism. Okay, now chemical mutagens, which are in a different class than radiation mutagens, come from direct exposure. So a chemical gets into a cell, or exposure from the environment involves absorbing or somehow taking in a chemical that causes damage to the DNA then internally. So nucleoside analogs would be the first case. A good example of one is 2-aminopurine. This is a nasty artificial chemical that is human-made and was discovered to look a lot like adenine. So here's your adenine here, normal adenine. Okay, and our cells look for this to incorporate to build DNA. And then you have 2-aminopurine over here, and it looks a lot like adenine. It still has a 5-membered sugar, which is going to be the deoxyribose. And then we see the nitrogenous base here. And it's similar, but not exactly the same. And that's what fools the cell. So the cell takes in the 2-aminopurine, which has this issue here where something isn't quite right, but the cell doesn't know better. And what happens is it says in the description down below that the AT, that is the normal pairing, changes and it becomes a CG pair as a mutation. So you can see how this could result in a base substitution or a type of point mutation. And so exposure to so-called nucleoside analogs like 2-aminopurine would result in a possibly lasting damage in mutation. Another type of chemical mutagen are nucleotide altering chemicals. This is where the bases that you have in your body that make up your A's, G's, T's, and C's can be changed by a chemical that comes in and alters them. So one major example that's well known is called nitrous acid, which has the molecular formula HNO2. And when cells are exposed to nitrous acid, it can result in a base pair substitution as well, like the previous example. But the way in which it works is that the nitrous acid makes contact with A, and it chemically modifies it. HNO2, nitrous acid, will modify the A so that it no longer binds with T. So A, which should bind with T normally, then ends up binding with C. And this is a very strange and unexpected mutation. Now, A will not stay bound with C long term. What ends up happening is once that C is present, the A may disappear over time and eventually become a GC pair over time. But at the end of it, it results in a base pair substitution. So AT effectively becomes GC if there's no way to stop it. And so there you have nucleotide altering chemicals. Scary stuff. A third type of chemical mutagen are what are referred to as frameshift mutagens. And if you remember, that was the insertion or deletion of one or more base pairs. And some chemicals can cause this to happen. Benzopyrene is an interesting example. This is a chemical that's found in smoke. and soot. I'm sure through decades of news about how bad smoking is, a lot of you can understand that there are many chemicals in cigarette smoke that can lead to cancer. Benzopyrene is found in that smoke, and soot is basically the carbon powder, the black tar that results from burning of items such as cigarettes. And then when this gets into your lungs, it can act as a frameshift mutagen by inserting itself in between the bases and causing a change in the reading frame. One toxin that or a chemical mutagen that you may not have heard of in this class is called aflatoxin. And aflatoxin functions very similarly, but it's found in moldy peanuts. And I imagine nobody really goes around thinking, I'm going to find some moldy peanuts to eat. But even in very small amounts, where peanut butter could be made or peanut-containing products, if some moldy peanuts are found in that crop and are mixed in, even to where it's just a small amount, a little bit can go a long ways here. The mold, in particular, comes from an organism called Aspergillus flavus. Aspergillus flavus is where afla comes from. You've got A and then FLA, afla. And aflatoxin is a well-known liver carcinogen, has caused many cases of liver cancer as the aflatoxin is converted into its active form when the liver tries to detoxify the moldy peanuts. In this section of the genetics chapter, part 2, we have many different mechanisms of genetic recombination, which recombinatorial events are referred to as any events that have a gene. So the DNA changes somehow, and one of those events is through vertical gene transfer. So a parent cell, like we see at the top here, will, from parent to offspring, transfer its genes vertically downward to the offspring or daughter cells. Other possibilities for genetic recombination come in horizontal gene transfer events. So this is where a transfer of genes may occur between the same generation of cells. And so there are 3 horizontal gene transfer possibilities. First possibility that we'll cover is transformation. Transformation is the uptake of naked DNA. A second event that we'll cover is conjugation, and this is a sexual transfer of DNA between 2 bacterial cells that actively do a connection with the sex pilus. And then lastly, we have the viral transfer of DNA from one cell to another, and that's referred to as transduction. And viruses that infect bacteria are referred to as phages, and so phage viruses will do this transduction as a means of horizontal gene transfer. So in this section, we'll look through each one of these possibilities as a way for genes to change in bacteria. And there are ways in which bacteria then can evolve and become something different than they once were. This is a particular figure taken from your book just for reference. So feel free to refer back to this if you want to summarize these fairly quickly. But we're going to go through these in detail in the next few slides. Let's look at transformation then first. So this mechanism of horizontal gene transfer involves a bacterial cell dying. So somewhere out there, or maybe somewhere in there, like in your gut, you can have, trillions of bacterial cells, and a bacterial cell eventually dies. Remember, bacteria may only live 2 to 3 days, so there's a lot of turnover where bacteria are dying each day, and as they die and their cell ruptures, it may release DNA. When the cell breaks apart during lysis, some of that DNA will come out in little fragments, and then that DNA potentially could be useful to living cells that, if they can pick up that DNA and take it in, then the recipient cells, as they're referred to, can change how they behave. And they're said to be transformed then. So a recipient cell, when we think about our laboratory experiment, these are referred to as competent cells. A competent recipient is a bacterial cell that can take up that DNA from the dead donor. And once that DNA is taken in, there's a chance that one or more sets of genes could be adopted or recombined into the recipient's genome. And so then this can bring about many changes to a bacterial cell. Some cells are naturally competent and can do this according to their natural mechanisms. In laboratory, we did a transformation experiment where we had to chemically treat the cells using calcium chloride, which allowed them to take up the DNA by us, using a chemical treatment to destabilize their membrane. So if you can recall back to that transformation experiment, that was an important way to understand transformation. Here's a summary of it. Transformation: naked DNA transfers from dead donor to a living recipient that's described as competent. It's important to conceptualize that competent cells are recipients that are usually young, actively growing and dividing, and or have receptors. Generally they would have to have a mechanism for taking that DNA in. And we can say that donor and recipient are usually either of the same species or very closely related, genetically speaking. And so transformation has profound consequences. Bacteria can evolve to produce certain virulence factors by picking up that naked DNA. Remember, virulence is disease-causing ability, so that could be the ability, maybe a newfound ability to make Fimbriae. Or capsules. What about capsule production? Or perhaps antibiotic resistance. Or lastly, toxin production. But why would a cell limit itself to just one? Sometimes transformation can result in an uptake of more than one of these types of virulence factors. I see that antibiotic resistance was also listed here additionally. So you get the idea. Transformation can bring about some profound changes in bacterial cells. Transformation was first discovered and described when a man by the name of Frederick Griffith performed a famous experiment, now known as Griffith's experiment, all the way back in 1928. He was researching a possible vaccination for the most common form of bacterial pneumonia caused by an organism called Streptococcus pneumoniae. Griffith was growing Streptococcus pneumoniae on plates, and when he looked at the colonies, he described them as having a smooth appearance. And this is due to the fact that Streptococcus pneumoniae covers itself, it surrounds itself in a capsule like we see here. And these encapsulated bacteria are known to be antiphagocytic. They can defeat the alveolar macrophages of the lungs and cause fluid buildup, sometimes to the point of a deadly disease. Griffith had taken these encapsulated Streptococcus pneumoniae cells and injected them into mice. And when these cells were injected into the mouse was overwhelmed and its immune system failed to be able to eliminate this pathogen, and so death resulted. Griffith performed a second experiment with a different plate of cells that he described as rough. Interestingly, these cells were the same species, still Streptococcus pneumoniae. These Strep pneumoniae cells, however, were mutant in the fact that they did not have their usual encapsulated protective structure. So Strep pneumoniae without the capsule we'll refer to as unencapsulated. Not enough room there, unencapsulated. Were injected into the mouse, and the mouse lived because the mouse's immune system was able to recognize and defeat this form of Streptococcus pneumoniae. In a third part of Griffith's experiment, Griffith took the normally dangerous encapsulated smooth strain of Streptococcus pneumoniae, as we see here, but he heat killed the cells. If you heat the cells up enough and denature them, then they will die. His hope was that if he took these heat-killed Streptococcus pneumoniae cells and put them in a syringe and then injected the mouse, that the mouse might have an opportunity to build up some immunity and also avoid a deadly scenario. In this case, the autopsy revealed that there were no live cells, so no pathogens were found in the plate because the ones that were injected were dead to begin with. Where Griffith's experiment becomes quite famous and revealed a new discovery, led to a discovery that was very surprising, was when Griffith got the idea that if he mixed heat-killed encapsulated organisms, so heat-killed smooth, along with live rough cells, that this mixture would be something that perhaps would do a good job at making for a form of a vaccine where you have a killed pathogen, but then you also have some live non-pathogenic or rough strain Streptococcus pneumoniae. But much to his surprise, this mixture killed the mouse. And even more fascinating, after the mouse was dead, the autopsy showed that only live smooth bacteria were found in the plate, no rough. And this was a big mystery because initially heat-killed smooth cells were injected. So they were dead, they shouldn't have been alive, and they weren't. But yet, at the end of the autopsy, the mouse had produced from its blood only live, smooth pathogens. You have Streptococcus pneumoniae. And so this was actually a way in which transformation was revealed to be a real phenomenon, whereby the heat-killed smooth cells had released DNA. Part of the DNA were genes for capsule production, and that those genes for capsule production had been taken up into the live rough strain. And that live rough strain gained a newfound ability to become capsule producers. And that allowed them to defy and defeat the mouse's immune system. And so transformation occurred in this case. And this is what Griffith's experiment had revealed had happened, was that genetic material could be transferred from a dead donor to a living recipient. A second type of horizontal gene transfer is called bacterial conjugation. Bacterial conjugation involves a living donor that then copies its DNA and transfers it over to a living recipient. One we would refer to as a gram-negative arrangement here through the sex pilus. So gram-negative cells that are able to undergo bacterial conjugation will produce a sex pilus if it's what's called F positive. An F positive cell, the F is in reference to fertility, the fertility factor. And for short, that fertility is referred to as F, fertility factor, or F factor as in for short. And when an F positive cell which has the F factor expresses this gene, it will start to extend this sex pilus out into its external environment, and a so-called F minus cell will act as a recipient. And this is a one-way street. The recipient receives the DNA that's donated over from its F positive donor. So the F positive is the donor. And this is a very important way in which bacteria may be able to then share genes across the same generation. In this picture on the left here, we can estimate, maybe take a guess, that the donor has fimbriae. We can see the long spiky protein structures here, fimbriae, used for attachment in the environment. And then the F minus cell may be receiving genes for fimbriae production across the sex pilus. So after this act of conjugation, it may be later observed that the F minus recipient could become able to produce Fimbriae, as well as maybe receiving a copy of the F factor. And in the next slide, we'll see what those possibilities are, where an F positive cell can transform, well, in a sense, cause an F minus cell to become positive. This is a arrangement in which a sex pilus is not produced, is not present, but conjugation can still occur, in which the donor and the recipient line up alongside one another and form a so-called mating bridge. The mating bridge does not require the pilus, but nevertheless, DNA can be copied from donor to recipient. This is still an act of conjugation, but via a mating bridge, as opposed to conjugation in gram-negatives, where we see the sex pilus, which will act as a transfer setup. Here's a figure taken from our textbook showing how conjugation can occur in E. coli. And so in E. coli, on the left figure here, we see that the F positive cell is able to make a sex pilus, has sex pilus production. And so the F factor can be copied over from the donor to the recipient. That's referred to as F minus because initially it lacks the F factor. Lacks the F factor. But after conjugation occurs, where genes are transferred over from donor to recipient, you can end up with the F minus cell being transformed into an F positive cell. So this is one result of how conjugation can change a recipient cell so that it evolves, so that it becomes something, it becomes capable of something that it was not capable of previously. Conjugation is an act that happens between two living cells. Different types of genetic information can be transferred over. Plasmids, which are referred to as extrachromosomal pieces of DNA, small little elements of DNA that can exist in addition to the normal chromosomal DNA can be transferred. Also, pieces or portions of the chromosome may be copied or transferred as well during conjugation. The F factor is responsible for sex pilus production, and it may or may not be transferred during conjugation. So this is something that if the F factor gets copied over completely, then the F minus recipient can become F positive. If it does not get copied over completely or at all, then the F minus cell can remain F minus, but maybe still receive some other important genetic information. So there isn't a rule that says that the F factor must be copied over in all instances. And so in those instances where the F factor fails to be copied over, there's a bit of an unusual event that accompanies this in many cases. And so we see here in this diagram how this would work. This little circle here is a plasmid. A plasmid is a little extra circular piece of DNA that's not part of the chromosome. And this F factor, this plasmid has the F factor on it. And so this F positive cell may experience a change at some point during its life where, for reasons that are not always understood, the plasmid may join, like we see here. The plasmid can become part of the chromosome and insert itself. When the F factor inserts itself into the chromosome, it becomes an integrated F factor. And a cell that has an integrated F factor is what's called an HFR cell. So HFR means high frequency of recombination. So to explain what that means, high frequency of recombination, first of all, it's the status that we see here. An HFR cell has to have an integrated F factor. And so then, why is it high frequency of recombination? We'll look at the consequences of having the F factor integrated. So that'll be in the next slide, but let's make sure we get this definition. Integrated F factor. Remember, integrated means into the chromosome, into the chromosomal DNA of the F positive organism. So here we see that an HFR cell, which is still F positive, right? HFR cells have the F factor, so they have the ability to make the sex pilus. So they do, and in that instance, when they hook up with an F- recipient, the HFR cell has to decide what part of its chromosome and or F factor it's going to copy over. And most often what happens is the part where it starts to copy seems to break apart right in the middle of the F factor. And so what happens is a cut happens right here. This is called the origin of replication. And in this origin of replication, it's then making it difficult to highly unlikely that all of the F factor will be copied over. And you might be wondering why that would happen, but we don't always know. So it's just something that's observed to happen, is the HFR cell will cut in the middle of the F factor there at the so-called origin of replication, and then begin to copy over DNA. So part of the F factor and neighboring genes next to the F factor will be transferred. And as they're copied over and transferred, then the recipient cell gets an interesting package. It can have any number of different genes that could be advantageous to making the F- cell newly capable, right? That's what recombinant means here, is that the F- cell may have, new characteristics. So things like toxin production or capsule production, antibiotic resistance, etc. But this recipient cell will not have received the entire F factor, so therefore it's still F minus. And that's just what ends up happening. So unusual event, and but we do observe that this does happen. And so it's a way in which bacteria can evolve by sharing genes. So we have HFR mating with the F minus recipient that stays F minus, but can still get genes that give it a way to change over time. So in summary, conjugation involves an F factor that might incorporate into the DNA, and if so, the cell becomes classified as HFR for high frequency of recombination. F minus cells stay F minus, but they may have received new genes, so they are referred to as recombinant F minus. This is because they didn't get the whole F factor, so they're not F positive, but they still evolve. And so we see a variety of virulence factors, which might include, just like earlier when I mentioned in transformation, things like capsule production, Fimbriae, toxins, or maybe antibiotic resistance. There's no limit to what these cells could receive. A third type of horizontal gene transfer is transduction. And transduction is carried out by bacteriophages. Bacteriophage are oftentimes simply referred to as phage or phage viruses. And these viruses are known to specifically attach to bacteria. So here's a little phage virus here. And what that phage virus is doing is it's attaching to the outer portion, the cell wall of a bacterial cell, and then successfully injecting its DNA into the bacterial cell. And ultimately, the mission of any virus is to take its genetic material and reprogram the cell to make more phage viruses, or whatever virus it is. They just want to make more viruses. So they reprogram the cell. And that's the normal cycle of a phage. The reprogramming is in an effort to make new phages. And these newly formed phage occasionally make a mistake. Something unexpected can happen where the new phage may package bacterial DNA instead of its intended viral DNA. So new phages may package bacterial genes. And these genes or DNA from this host cell then could be transferred to a future recipient. And so when the host bursts open with a whole bunch of newly formed phage, one of these phages might just pick up some bacterial genes from this recipient, from this previous host, and inject it into a new recipient. Now at this point, this recipient is pretty lucky, because instead of getting the genes for phage viral infection, perhaps a happy accident can occur here. And we say that because the recipient cell is now getting bacterial genes from elsewhere, without the downside of forming a viral infection. And so what we can have here, at least in this particular diagram, is a cell that picks up new genes and becomes recombinant and able then to gain new abilities. So these cells down here have changed. They've evolved through an unintentional action of a phage virus. So recombinant cells result. Transduction, in a general summary, involves a virus known as a bacteriophage, or simply phage, that is able to move DNA. And one way of terming that is it acts as a genetic vector. So it's picking up DNA and passing it from a donor initial host over to a future recipient host. So the donor DNA, if transduction is successful, can then incorporate into new recipient DNA. The donor and recipient are thought of as being in the same species, or maybe even in the same subspecies or strain, because bacteriophage are very specific to their host in the vast majority of cases. There are some rare instances where a bacteriophage could adapt to infect more than one species. Those are so-called promiscuous phage. That's kind of a funny term for them, where things can get a bit fast and loose. But in most cases, we say that the same species are involved, or even the same subspecies, so even more specific than just all of one given species. So phages, when they do this active transduction, we can further specify that there are two major types of transduction events. There's generalized transduction and there's specialized transduction. The one that I went over in the previous slide was a model for generalized transduction. Generalized transduction, as it was outlined a couple of slides ago, describes the way that transducing phage operate when there is a random transfer of any genes. So a donor is infected with the bacteriophage, and then fragments of the donor bacterial's DNA is surrounded by the virus's capsid. The virus capsid is a protein shell that wraps up the genetic material for the virus, and then the virus can carry the DNA over to a recipient. But before that can happen, the donor has to lyse after a load of viruses are reproduced. An infected bacterial cell could release anywhere from 50 to a couple hundred new viruses, and some of those viruses that are released, if they happen to have packaged up bacterial DNA, they can take that donor DNA, deliver it to a recipient, and then if that combines, then you have a recombinant cell. However, there is a chance that this process could fail. And so if a donor cell fails to adopt the new DNA that it's been given, and this is what's called abortive. So the recipient cell may not actually receive the DNA and successfully change by recombining that, and so it may destroy the DNA. But if in fact transduction is successful, then that transduction event could involve the transfer of many virulence factors, like mentioned in the previous slides, ranging anything from capsule production to Fimbriae, to the ability to produce one or more various toxins and or things like antibiotic resistance. And so many different genes may be transferred over during a generalized transduction event, but it's done so in a random nature. A second type of transduction is called specialized transduction. And specialized transduction is, as the name implies, only specific genes will be transferred. Okay, so we're saying that phage viruses get in, but when they pick up bacterial genes that they may carry to a new recipient, only specific genes will be transferred during specialized transduction. And this event is a bit more complicated because the less than random nature of things involves something called lysogenic phages. So lysogenic phages, as they're sometimes called, produce what's called a mild infection or a silent infection. Temperate, if you were talking about the weather or the climate, would mean an infection with a lysogenic phage is one where the bacteria may have a period of silence. And so the way that this works is that the donor that gets the viral DNA from a lysogenic phage, okay, so lysogenic phage DNA goes in, but then instead of just taking the cell over and producing more phage viruses right away and causing the cell to burst, instead the viral DNA goes in and it integrates, it incorporates in to the bacterial DNA. So bacterial DNA combines, or I should say fuses, with the viral phage DNA. And this is referred to as a prophage. So a prophage is the term that we use to state that a bacterium has fused its DNA with the phage DNA. And during this time, we get what's called a period of silence or lysogeny. So lysogeny is a time of waiting where the virus is there, but it's not active, it's not doing anything. So the virus is inactive. Or silent. But after some amount of time, the virus may become active again. Something triggers it. It's not always known what these triggering events are. To review this, to clarify, you have a phage, and not just any kind of phage. We'll call it a temperate or lysogenic phage. And what this temperate or lysogenic phage does is it infects the bacterial cell. So infection occurs. And then that host will undergo what's called lysogeny. And lysogeny is a waiting period. It is it's a silent infection. And for how much time we don't know. It could last generations, actually. So this bacterium could grow and multiply several times, all the while having this dangerous viral DNA in there. But following some sort of trigger or activation, then the final stage would be an active viral infection again. And so this is referred to as the lytic cycle. The lytic cycle is where the virus becomes active and the cell will burst open. And this lytic cycle will take with it phages that only transfer specific genes. So the way that this happens is that the viral DNA, when it pops out of the donor DNA during the activation or the triggering event, is that the neighboring donor DNA is next to the viral DNA. So the viral DNA, as it pops out, takes a little bit of the neighboring bacterial DNA. And so this can transfer very specific genes depending on where the viral DNA was inserted. So specialized transduction ultimately, just like other horizontal gene transferring events, can involve the transfer of capsules, fimbriae, toxin-producing genes, antibiotic resistance, you name it. All right, so one thought question that came from our book asks, E. coli, which we know is found naturally in the human large intestine, is a part of our normal flora. It's beneficial, but we know that there are dangerous strains of E. coli. So we've all heard about reports in the news in which E. coli has caused foods to be contaminated and pulled off of shelves and out of grocery stores nationwide, if not, on a global scale, mainly because of agribusiness, the production of foods, whether they be dairy products, where there are cattle. The cattle also have E. coli as normal flora. But then there are strains that we're not familiar with in the human body. They're foreign to us. And one really dangerous strain is the E. coli designated O157H7. And one of the ways in which we know that this particular deadly E. coli is different is in its production of something called Shiga toxin. And Shiga toxin causes bleeding and major trauma to the large intestine, down in the colon and large intestine area. This toxin will cause bleeding, and then E. coli can get into the blood and cause a septic episode. What we do know about Shiga toxin is that its name stems from the fact that it comes from a entirely different pathogen called Shigella. And Shigella is a diarrheal pathogen that's well known to cause deadly diarrhea and septic shock. But it's supposed to be found in Shigella, not in E. coli. So when it was discovered that this unusual variety of E. coli called O157H7 was found to be dangerous because of this virulence factor that it had picked up, called Shiga toxin, well, the big question was how did it get it? So how did E. coli acquire the gene from Shigella? You might be surprised to learn that the answer is from a transducing phage. So an unexpected event occurred in which transduction occurred from a bacteriophage that originally was able to infect Shigella, but then somehow that phage became less specific and had the ability to take genes from Shigella, which were undoubtedly in the gut of an infected person, and then float over to some of the normal E. coli, and then transfer those genes over. Whether that happened in an animal, like a farm animal, like a cow, or a human, we really don't know. But a transducing phage was discovered to be responsible for this event. Although it's unexpected, it's an interesting development that geneticists were able to figure. In the remaining handful of slides, I'd like to explain plasmids and transposons as genetic vehicles, pieces of DNA that are additional. They may not occur initially within an organism's genetic package, and they can move around, and they can contain elements that are above and beyond what a basic chromosome in a bacterial cell or other living cell might contain. So a plasmid, by definition, is considered additional genetic material above and beyond the chromosome. So another way, plasmids are extra chromosomal. And plasmids are not just limited to bacteria, which are prokaryotic. They've also been discovered to be found in many types of eukaryotes as well. And plasmids, when they exist independently of a chromosome, are found to be small circular pieces of DNA. And plasmids are interesting in another respect as well, which is that they have the ability to direct their replication in a self-directed sense. They self-replicate, and they can exist in high or low copy number. And this is just a way of referring to the fact that sometimes the numbers of a given plasmid can increase to quite high numbers. A high copy number plasmid may replicate itself dozens of times. There could be 50 copies of a plasmid floating around inside of a cell in a high number. However, there are times when plasmids can exist in just one copy or a few copies, and so they would be considered low copy number plasmids. Plasmids are influenced by their genetic sequences to self-replicate. That subject goes deeper into genetics beyond what this course is going to have time to address, so I'll leave it at that. Plasmids are also considered to be non-essential pieces of genetic information, but that doesn't make them unimportant by any means. Plasmids have been found to make all the difference in some cases with respect to highly resistant infections, so-called R plasmids. Right? R plasmids, the R is in reference to resistance, and so R can refer most often to resistance to antibiotics. Here is one particular plasmid with a so-called R factor. This is a picture taken from our book. On the right, it shows a lot of different parts of the plasmid, ranging from an origin of replication where it can be copied. There's an area, if we go clockwise from there, where we can see mercury resistance. We can see genes for things like sulfonamide drugs, sulfa drugs, which are a major class of antibiotics. Streptomycin, a different type of antibiotic. CML is for chloramphenicol. And so here we have a whole portion of the R plasmid that it has a cassette of antibiotic resistance genes. TET, that's for tetracycline down here. And interestingly, on the other side of this plasmid, there are genes for the F factor. If you remember, the F factor forms the sex pilus. F stands for fertility, and so the F factor has the instructions that give rise to the sex pilus and conjugation. So we know these plasmids exist. This is an example of a sequence of one such plasmid. We can see in this picture here on the left, every one of these loops found in this picture of the cytoplasm taken really close up. This has been magnified millions of times and is a scanning electron micrograph of the cytoplasm inside of a bacterial cell, and it's showing various plasmids. Every single one of these loops is a plasmid. They're all plasmids. This is a high copy number plasmid, as we can see. So plasmids can be transferred over during transducing events, during conjugation, even during transformation. Another type of genetic vector are what are called transposons, and a vector, if you recall, is some sort of mechanism that carries genes, a genetic vector, and in this case transposons are referred to as jumping genes. Like plasmids, they are small segments of DNA. They can exist in a circle or a loop, but they are not referred to as plasmids necessarily, because they were discovered to behave more loosely than plasmids, to have more possibilities than a plasmid. They were first discovered by a scientist in the 50s who, by the name of Barbara McClintock, and she was studying how corn kernels could have different colors, and that the pigments that gave rise to the different colored corn kernels was actually due to the fact that transposons had allowed genes to jump around inside the corn plant and produce these different colors. And transposons since that time have been found to move from one part of a chromosome to another, so on the same chromosome, or even more unusually, to different chromosomes, or even onto plasmids. So there really are no rules, it seems, for these transposons. They can jump around, and they also can move without replicating. So they can copy themselves and move, or they can just pick up and move and not replicate before such an act. They have been found in all types of cellular life, from prokaryotes to eukaryotes to even viruses. So transposons are kind of a discovery that breaks all the rules, so to speak, as to where genes can remain. It shows us that evolution has possibilities, at least through transposons, to move genetic material from places that we never thought could jump from one place to another. So as it says here, transposons can jump between unrelated species as well. And one of the most problematic discoveries within the world of transposons was when it was discovered that a very problematic type of antibiotic resistance called vancomycin resistance had transferred itself from the infamous VRE, which stands for vancomycin resistant enterococcus, which has killed people for many years, decades really, since the 1980s, in hospital patients, that form of genetic resistance, antibiotic resistance, had been transferred over to Staph aureus. Vancomycin, previous to this event, was a fairly reliable kind of last line last resort drug for Staph aureus patients, staph infection patients who had had MRSA problems, methicillin-resistant Staph aureus. We now know today that roughly 25% of MRSA patients, of staph infection patients, have methicillin-resistant Staph aureus. And so it's much more common that we rely on vancomycin antibiotic to treat those staph infections. Unfortunately, not all of those patients will respond to vancomycin therapy, and it seems in large part due to this event where transposons have brought vancomycin resistance to Staph aureus. Here's a figure just visually giving you an idea of all the possibilities that transposons have in their existence within cells. So you can see here, looking from one area to the next, this is where the transposon is. That transposon could move to a different area on the chromosome that it exists. It could copy itself and then move on to several locations on a chromosome, or transfer to a different chromosome entirely. Or in this case, finally, we're seeing that the transposon could just excise itself, cut itself right out of the chromosome, and then move on to some other place. So these are the possibilities that transposons have, and it gives us a real eye-opening perspective on how bacterial evolution, genetic evolution in maybe any life form could be influenced by the presence of transposons and their movement through various species. So we have a final concept check, and it's asking the question, which of the following mechanisms of horizontal transfer involves the transfer of bacterial DNA through a bacteriophage? So we'll just skip right to it. A bacteriophage has to be involved in what's called transduction if it's moving genes around. But let's make sure that we understand why the wrong answers are wrong. First of all, transposition is involving transposons, but not necessarily specifically bacteriophage. So we would not pick that as the one best answer. Conjugation, if you guys recall, is where living two live bacteria hook up in a sexual act, and DNA is copied and transferred over from donor to recipient. And transformation is does not apply because a dead donor has to just release its DNA, spill it out into the environment, and a living recipient has to pick up that DNA, a competent cell, and no phage has to be involved there. So the answer is transduction. I hope you guys are able to recall that easily at this point. And finally, we have a slide about just the idea of genes and evolution in general. When studying genetics, it should be kept in mind that organisms change over time due to all the mechanisms from mutation, whether it's random or through events that expose cells to mutagens or chemicals, environmental stresses. We also know then that recombination can occur. So recombination can be these vertical gene transfers from parent cell to daughter cells, and then you can have all of the horizontal gene transfers, right? Transformation, transduction, conjugation, and so you get a lot of movement of genetic information and evolution from that. And lastly, just it's sort of a Darwinian outlook here, right? Survival of the fittest. Organisms that are best suited for their environment will be naturally selected for. The ones that have the genes that allow them to survive for longer are the ones then that we're going to see more and more in the future, whereas the ones that are no longer fit will die out. And we know that no matter how many antibiotics we come up with to try to defeat the bad guys, they do have these sneaky ways of mutating and recombining their genes with their neighbors. And so through a process of natural selection, the fittest organisms with the resistance factors, they will stick around, and they will eventually become plentiful if we keep the same strategy of trying to control those organisms. And that is it for genetics, everyone. And the remaining slides are slides that are just for reference. If you have a little reading that you'd like to do that's just taken from a textbook that could give you a little depth and understanding. So the appendix has some long descriptions. Feel free to read those and pause on those if you would like some deeper reading taken from your textbook. All right, thank you very much, and we'll see you for the next lecture.